afternoon, everybody. Thank you for coming today. Should I check Princeton?
I think Zoom's probably fine. Jesse's
Okay.
monitoring it,
All
so I
right.
think you're good.
All right, so thank you for here today. I'm very happy to introduce today's guest speaker, which is Dr. David Van Sanford, as you know. But we have new graduate students and maybe some new staff members. So I would like to introduce him. So he got his bachelor's degree at Oregon State University. Then he went to Colorado State University for his master's degree. Then in 1981, he got his PhD in genetics at North Carolina State University. So 1981 is the time he came to the University of Kentucky. Since then, he's here. He's now a professor, working on several things, and then today he's going to talk about some of his research projects. Along that, he got several awards, and he's the fellow of American Society of Agronomy and the Crop Science Society of America in 2011. And he also got several awards from University of Kentucky, Thomas Paul Cooper Distinguished Achievement in Research Award 2014, and University of Kentucky Research Professor Award in 2018. So it's very pleasure to have him today to hear about his research. Thank you.
Thanks, Tomo. Howdy, everybody. Good to see you all here. Appreciate the invitation to present. So what I want us to consider today is the question, what is the role of plant breeding in elevating the climate resilience of our agriculture? And I must confess that this question has been rolling around in my brain for the better part of a decade in one form or another. And it really traces back to an event that our department hosted almost exactly a decade ago. And this was in response to stakeholder interest from the Kentucky small grain growers. It was the Climate Change and Small Grains Production Workshop. And so we had some guest speakers in. It was a very productive conference. It led to some long-term collaborations with Stu Foster, for example, who was the former state climatologist at Western. And it was the first time that I had met Sanford, who I consider the preeminent wheat modeler. And so it was a little less than a year later that I went down to Sanford's lab and spent a 6-week study leave, hanging out with him and some visiting modelers, kind of learning how they thought about things, and starting to think about this whole question in earnest. So if we're going to talk about plant breeding today, I need to emphasize that plant breeding is a cyclic enterprise, and it's often depicted as just a static circle. But I think it's important that we include the element of time, the dimension of time here. And I've put another couple of circles on here to indicate its recurrent nature. So basically, we make crosses. From those crosses, we create genetic variation. We select among the progeny of the crosses. We extract some breeding lines. We test the heck out of them. And then if we're extremely lucky, we end up with a new variety to release for farmers to grow. And the new varieties and the breeding lines, the elite breeding lines, become the parents of the next cycle of crosses. All this time, we're moving forward in time, moving along, and it's this fact that gives rise to what I call the business as usual hypothesis, which holds that because we are forward facing and we're moving on in time and we're practicing plant breeding in a warming climate, we therefore must be bringing along genes that confer climate resilience. This is the assumption. In fact, we really don't have a good idea much on what genes we are bringing along with each cycle of selection, except for those few big ticket genes that affect plant growth habit, like photoperiod, height, and so on, or disease resistance genes, all of which have been tagged with DNA markers so we can track them. But beyond that, we don't really know. And so I'm reluctant to embrace this hypothesis, really, because I think that we can't assume that we are bringing along those genes. And I happen to believe that an overt effort is needed in addition to what we're doing in the business as usual model. And I just want to mention something here that you can kind of put in your back pocket when we think about genes and populations and moving through time. We can just say simply that the goal of selection is to change gene frequency. So what we would want to do in this kind of an instance is to increase the frequency of those genes that confer climate resilience. So just to give you a little sense of one reason that I don't really embrace this hypothesis fully, this data was kindly supplied by Matt Dixon, and I asked for some data from 2007 to 2021. So our most recent variety release, Pembroke 2021, the initial cross was made in 2007. It was a three-way cross, so then we took that first F1 and crossed it to another parent the next year. And after that point, we took the segregating populations out into the field and did selection. Now, it was during this period that in 2012, we had a hellacious spring freeze. And in 2014, we had our first taste of the polar vortex. So I can't assume that the population that gave rise to Pembroke 21 was subjected to selection pressure for that would confer success in a warming environment. Okay, so since we're talking plant breeding, and on the off chance that you haven't been thinking about genetics recently or plant breeding, I'll just do a quick run through some of the terminology that we use and that I'll use today. We speak of an allele, that's one or of two or more versions of a gene. Phenotype, that's the data that we collect in the field or in the lab, like grain yield, protein concentration, low volume, what have you. Genotype, the genetic constitution of an individual, depicted here at these three loci. Heritability is that proportion of the variance that we measure in the field or in the lab that we can attribute to the direct effects of the genes. And finally, genetic gain, that is our response to selection. So this so-called breeder's equation says that genetic gain, delta G here, is a product of heritability and selection intensity. So selection intensity, here we are selecting out in the tail of the distribution. So fairly intense selection. If we have a high heritability, we're likely to have a good gain. All right, so what I really want to talk about today is the impact of a warming climate directly on the plant. But before doing that, I want to briefly speak about another consequence of climate change that certainly is very important and deserves a mention. So this publication that was, the first author was Katie Russell, a grad student at the time. And it came out of the same event that I talked about earlier in 2012. And Katie did a very thorough literature review at the time. And on this topic, basically, these bullet points say it all. Changes in temperature will likely cause shifts in pest populations and severity. And earlier springs and warmer winters could very well give rise to higher survival rates of disease-causing pathogens. So in this context, let me just give you three examples of things that we might face in Kentucky. So here we have pictured the wheat rust. So from left to right, leaf rust, a huge problem out in the Great Plains. Stem rust, globally, a big problem. We see leaf rust at some level every year. We very rarely see stem rust. And over here is stripe rust, which is historically confined to the Pacific Northwest, but there's a new variant that functions at higher temperatures that we see occasionally. So this is one that we're concerned about because Katie's point about the warmer winters, right now we don't see leaf rust in a damaging way every year, because it arrives too late in the crop cycle. The spores blow in from Arkansas and Louisiana, and they don't do enough damage to warrant a lot of attention. That could very well be changing. Okay, this is a more nefarious pest by far, wheat blast. Been a real problem in South America, started in Brazil, moved away from the equator, south into Bolivia, and then into Argentina. Its symptoms look amazingly like head scab, and like head scab, it prefers warm, wet conditions. So if we were to fast forward our environment in Kentucky, N years, I don't know what N is, and maybe all of a sudden our climate looks more like Bolivia than it does now, wheat blast could be a problem. And this is, it's a devastating disease. Resistance genes are few and far between, unlike the ruts, where there are fairly abundant resistance genes. So this is something that we don't want to see. And then finally, Hessian fly. So in the 40 plus years that I've been here, I've been waiting for the other shoe to drop on this critter, and it never has. It is potentially a very devastating pest. It arrives in the fall, lays eggs, the pupal and larval stages occur within the plant. Affected plants are severely stunted, yield is severely reduced. Now, we do have resistance genes, a fair abundance of them for this insect pest. Not a problem for us, but it's a big problem south of here in North Carolina and in Georgia. Hessian fly has several generations a year, and so it's a big focal point for them. So all of these are problems that we know how to deal with. They would require a shift in resources. And right now, talk about the effect of a warming environment on the plant. So this is a paper, by now it's got some age on it, from the aforementioned Dr. Aseng and company, which predicted, according to modeling work, that for every 1 degree C increase in global mean temperature, wheat production could decrease from 3 to 8%. And the big impact would be grain yield and grain number, not so much grain size or harvest index. So yes, we know that wheat yields are going to be reduced by heat stress, but we also know that increasing levels of CO2 are going to offset this to some degree. But that also has a negative consequence, because we'll likely end up with lower protein levels simply via a dilution effect. More carbon, less nitrogen. Not a nutritional problem in our advanced society, our wealthy society, but it could pose a significant problem in certain parts of the world. And then this recent paper, which was one of the few to really try and weave plant breeding into the story. So they are predicting by mid-century, just about a 2% reduction in global wheat production, with up to 15% in Southern Africa and South Asia. So that's depicted here in this first part of the graph. And then they say, okay, if we focus on a certain set of traits, and their traits are like early vigor, early water use, and late flowering, then we could mitigate that to some extent. And then if in the parts of the world where nitrogen fertilizer is not abundant, we add nitrogen fertilizer, then we could even make a much bigger difference. Okay, so to select for climate resilience, there's a few things that breeders would have to do. The first of these would be to determine the sources of genetic variation, and then identify traits that we're going to screen and figure out some kind of a screening protocol. We estimate heritability of the traits, and finally, optimize breeding methods. So when we think about sources of genetic variation, it behooves us to recall the richly detailed and documented evolutionary history of wheat. The wheat cultivated wheat, Triticum estivum, is an allohexaploid species. Hexaploid meaning it has 3 genomes, allo meaning they're different from one another. So they come from 3 different species. So it's believed that about 10,000 years ago, the A genome donor pollinated Aegilops speltoides, the B genome donor, and spontaneous chromosome doubling occurred to give rise to this tetraploid wheat, Triticum turgidum. That's the precursor of the durum wheat that we grow today, from which we make pasta. And then about 8,000 years ago, that tetraploid was pollinated by this little guy, Triticum tashii, and the same thing happened. Spontaneous chromosome doubling, and that gave us ultimately the wheat that we grow today and the wheat that we cultivate. All right. Why am I telling you this? Why is it important? So to fully answer that, we have to mention the founder effect. The founder effect holds that if we have an existing population and we make a new population out of just a few members, then we're going to lose some genetic variation. And that's basically what happened here, because these hybridization events that I described were extraordinarily rare. And so many of the useful alleles in the wild species did not make it into the cultivated species. We know this not just because the founder effect tells us that, but because the wild progenitor species they've already been mined for many, many useful pest resistance genes, passion fly resistance, leaf rust resistance, scab resistance, and not just the ones I was showing you in that photo. I think 54 species have been mined to date. So lots of wheat has a huge potential gene pool. These might be a valuable source of genes for climate resilience, but it would be helpful if we knew something about the progenitor species and how they had behaved in a warming environment. And thanks to some very clever scientists in Israel, we do have a glimmer of knowledge there. So what these folks did, and this is just a figure from their paper, and they also look at wild barley, which I'm not going to talk about. On the x-axis, we have 10 populations of wild emmer wheat. So that's the tetraploid that we saw a minute ago, the AABB, that were growing in Israel. And they were sampled in 1980 and then again in 2008. And on the y-axis, we have days from planting to flowering. So what do we see across the board? Things are much earlier in 2008 than they were in 1980, right? And this is something that we've all seen in our daily lives. We see it in our backyards, I believe. So these guys also did some genotyping as well as phenotyping. Kind of a good news, bad news story. Quite a few alleles were lost. That is, alleles, like 73 alleles that were detected in 1980 were not found in 2008, but 45 new alleles were found. So that offers some hope for this idea of mining these progenitor species for valuable alleles that could confer some form of climate resilience. All right, so having set that stage, I would like to talk about some warming studies that we have carried out within the confines of the wheat breeding program. So the first study was conducted by Katie Russell, who I mentioned earlier, PhD student, and she carried this out over 2 growing seasons, screened 40 genotypes that offered various combinations of photoperiod sensitive genes. And so the B genome was just uniform across this set of 40. There was no variation, but there was variation in A and D. And it turns out that the D locus is the one that really determines what's going on for the most part. Per experimental unit, paired head rows, and you can see the head rows planted out here. 2 replications. The treatments were ambient and artificially warmed. And the warming was done with these warming cables that were buried just beneath the soil surface to heat the rhizosphere. We were aiming for 3 degrees C. That was our goal. And we used a Campbell data logger that was programmed so that if that difference between ambient and warmed fell below 3 degrees C, a warming cable was turned on. We had these thermocouple temperature sensors throughout the experiment, sampling both soil and air. I'd say about the lower 30 centimeters of the canopy were also warmed in addition to the rhizosphere. And the sampling interval was 15 minutes. So this is what we saw in terms of the spread between ambient and warmed over the 2 years of the study. We were pretty pleased with the job that these cables did. We had some soil moisture probes out, and we did not detect any impact of the treatment on plant available water. All right, so what did Katie see? Well, much like the folks in Israel that I just mentioned to you, both years of the study, she saw an earlying effect of the warm environment. And on average, over the 2 years, heading date was accelerated by 5 days. So in our environment, that's a significant difference. If you were talking Pacific Northwest or United Kingdom, no, 5 days would not be anything big. But in our environment, it's pretty substantial. Okay, and so what do we see in terms of impact on mostly agronomic traits? Yield, biomass, spikes per meter squared, nitrogen use efficiency, and total plant N, this would be like in kilograms per hectare, were all significantly reduced by warming. Nitrogen traits per se, it was kind of a mixed bag. We saw in 2015, virtually nothing was significant except protein, and a little variation. We saw some significance in 2014, but nothing real consistent. Now, to just focus on protein for a second, a half percent difference in protein can be significant. So this was not a trivial difference, but still it was not significant in the preceding year. So kind of an unknown. What interested me a lot was that of the 40 genotypes that we screened, 13 actually had a yield increase in the warmed environment. 20 genotypes had a biomass increase. Now, I mentioned the photoperiod genes earlier. So let me just talk a little bit more about them here. So we divided all of the lines up into whether they were photoperiod sensitive at the D1 locus or insensitive. So photoperiod sensitive lines only begin to elongate or bolt in the spring when the day length is sufficient. They're not driven by heat units. Photoperiod insensitive lines, though, are driven by heat units. The day length basically does not matter. And so we have a nice dichotomy here when we compare the two groups of lines. No significant impact on yield from warming in the photoperiod sensitive types. A significant reduction in yield, though, for the photoperiod insensitive types. Generally speaking, I'd say of the wheats that we tend to grow in Kentucky, the ones that show up in Bill Bruning's variety trial, for example, and in farmers' fields, probably 2/3 to 3/4 of them tend to be insensitive. So if we wanted to move everything over to photoperiod sensitive, it would be a big job, but it would be doable. All right, so this is kind of a busy, unattractive slide, I realize. Heritability estimates. I mentioned heritability earlier, and it's busy because we're including a 90% confidence interval here. So I'm showing you the lower limits and the upper limits of the confidence interval. these first two traits I've highlighted in blue because they're kind of our barometers when we do heritability. We expect them to be high. Height is a high heritability trait, generally speaking. Heading date is as well, and height certainly behaved itself. Heading date sort of middle of the road. But what's interesting is that over in the control environment, none of these traits really showed up as being heritable, i.e. the lower limit of the confidence interval was below 0. So that means they were not significantly different from 0. In the warmed environment, however, we did have significant values of heritability for all of these traits, the ones spikes per meter squared, yield, grain end content, and on down the line to end use efficiency. All right, so the bottom line for this study, warming will accelerate development. That's not a surprise, but we're glad to see that our system was working so that it worked out that way. And photoperiod sensitive genotypes may be useful in breeding for resilience. The heritability estimates argue that we should carry out selection under warming conditions rather than under control conditions. All right, so Katie also wanted to do a passive night warming study because of the importance of night warming due to photorespiration and so on. So we contacted the folks at the Cevieta Long-Term Ecological Research Unit near Socorro, New Mexico. Got some design ideas from them. They were really helpful and nice to work with. They gave us a source for this thermal fabric. So this came out and covered the canopy at sunset, and then it retracted at sunrise, daybreak. We were hoping for a 1 degree C difference between control and warmed, and we never really got there. We did not achieve the warming level that we were hoping for. I'm not saying we expected it, but we hoped for. Katie did see an effect on phenology, but it was far more muted and much less consistent than we saw with active warming. Not significant in 2016. It was significant in 2015, but the differences were really trivial, half a day's difference. We didn't see a great deal there. Yield, spikes per meter squared, both of those were significantly reduced under this modest level of nighttime warming. Biomass was not. And utilization efficiency, we got a reduction there. And total plan N, we actually had an increase under warming. Okay, so just to compare these two studies, yield and spikes per meter squared significantly decreased under both types of warming. Phenology was accelerated much more with active warming, and the relationship of nitrogen use efficiency traits to warming needs to be explored. So the third study was conducted by a master's student, Caitlin Hitz. Caitlin had a huge master's thesis. She had 5 or 6 studies, really ambitious, very much wanted to do something associated with climate change. And she was working on end use efficiency for the most part anyway. So she had the same setup that I've described previously for Katie. Now, she was using, among other things, a SPAD meter to measure chlorophyll as a proxy for nitrogen. At anthesis, no difference between the two environments. At maturity, significant reduction in the warm environment, no effect on biomass. But kind of a mixed bag on end metabolism. Nitrogen harvest index was significantly reduced. That's the amount of the total plant nitrogen that's captured in the grain. Utilization efficiency significantly lowered, and overall end use efficiency significantly reduced. pretty consistent effect on morphology and maturity traits. So anthesis state, as was seen earlier, significantly, and anthesis was significantly accelerated. Maturity date, physiological maturity, also plants were shorter, yield was reduced, and harvest index was significant at the 10% level. Now, Caitlin used a different set of material in her study. She was not looking at the same genotypes that Katie had looked at. So she used a subset of a big mapping population that had been genotyped with genome-wide markers, SNPs, single nucleotide polymorphisms. And she found that 3 of these SNPs associated with the chlorophyll index actually were significant and explained, when we see this level of explanation with a SNP, we're anything above 10% we're sort of interested in. So these were definitely of interest. And in the control environment, a SNP that was associated with N utilization efficiency that actually explained quite a bit of the variation. Okay, and then on to the 4th study. And I'm showing you this slide because it illustrates the kind of microplots that we had to use in Lisa Tessman's study. These are little hill plots. So, as you may know, we spend a lot of our time working on fusarium head blight or head scab in wheat. And infection occurs during flowering time. The anthers are usually the target. And its favorite environment is not unlike that I mentioned earlier for wheat blast. Consequences can be pretty dire. You end up with these bleached spikes, very shriveled grain, and usually they're loaded with this nasty toxin, deoxynivalenol or DON. And that causes problems in the feed and the food industries. Okay, so Lisa was using this big mapped and marked, genomically marked mapping population to do a GWAS or a genome-wide association study. So in this kind of a study, you've heard several people, Virginia Verdes among others in our group, talk about it. You look for an association between genomic regions and traits, and the genomic regions are represented by these SNPs that are scattered all over the genome. Okay, so our objectives in this study were to evaluate scab in this large, diverse mapping panel. In theory, it represented all of the germplasm in the eastern wheat growing region in the US. And to determine whether the resistance genes that we had come to rely on were going to be effective under warm conditions like they are under controlled conditions. Okay, big populations, 2 reps in 2016, 4 reps in 2017, same warming cables. Okay, in 2016, we were hoping that we could create a disease epidemic with a conidial spore suspension. That was not super successful. So in 2017, we went back to our old standby scabby corn, and you can see these black entities down here on the soil surface. Those are corn kernels that have been completely colonized by the scab fungus. Phenotyping, just the usual suspects were measured, but of real interest to us were the scab traits, scab rating, fusarium damaged kernels, and the toxin levels. And from a statistical standpoint, the only thing I'll mention of interest here is just this R package GAPIT that we use to do the genome-wide association study. Okay, so Lisa found, first off, significant genotypic variation for all of the traits she measured. Not a huge surprise because it was a big and a very diverse mapping panel. The treatment effect, the warming treatment effect, was significant for heading date. Again, we've come to expect that. And also for toxin levels and fusarium damaged kernels, not for scab rating, height, or yield. So impact of warming on heading date, this is much as we've seen in the other studies. things are much earlier in the warmed environment. Kernel damage is way worse under warming than it was under control. And to our knowledge, no one else has done this exact kind of a study. We sort of, the global predictions would argue that this is the likely result, that scab's going to be worse under warming environment. And toxin levels were much higher, significantly higher under the warm environment. And we did see some variation among lines and varieties. So this Purdue line, under controlled conditions, a low DON level, and under warm conditions, also very low. But this Ohio State variety Becker, pretty low DON under controlled conditions, through the roof under warm. So there is some genotype by warming environment interaction going on there. All right, so this is really the figure that you're looking for when you do one of these studies. On the x-axis, we have the 21 chromosomes of wheat, and you can see the A, B, and D genomes indicated. And on the y-axis, this is a log scale, and it's simply telling us the likelihood of seeing what we see being due to random chance. If we see some little dots above this line at 3, that's saying that the likelihood that that's due to random chance is less than 1 in 1,000. And so Lisa found that there were 3 disease-reducing SNPs that were discovered, but they were only expressed under controlled conditions. And that was for fusarium damaged kernels, and for DON, there were 2 disease SNPs, disease-reducing SNPs, again, only expressed in the control environment. So what happens if you get one of these and you validate it in another set of breeding lines, then you can develop a marker. Will any of these become markers? I don't know. They have not yet been validated in our breeding lines. Okay, so disease levels were higher in the warmed environment. GWAS identified several SNPs for reducing disease, but only in the control environment. Generally speaking, superior genotypes for DON under controlled conditions were often the best performers in the warm treatment. But selection for performance, we concluded, should be carried out in a warm environment. All right, so to kind of summarize these studies, did we see evidence that genetic variation for resilience exists in adapted soft red winter wheat germplasm? I think we did. We did observe the responses that are predicted by crop models. We see that we can expect increased scab levels in a warmer environment. But we were doing all of this as kind of an add-on to the breeding program. And it was a great experience for the students and for me, for all of us. But really, to do this well, to do it right, to understand the underlying genetics of resilience, we have to have a comprehensive, very well-funded effort. Now, I've only been associated with one large grant that had to do with climate change, the so-called TCAP grant, a coordinated agricultural project funded by NIPA. $25 million over 5 years, 50 plus PIs across the US. These were the traits that people focused on. Very productive grant in the sense that lots of papers were published, lots of varieties were released, lots of graduate students were trained, lots of educational products were developed and still reside on the web where they can be used in classes, and they are quite useful. Lots of worthwhile and enriching relationships with minority serving institutions were established. So that was all to the good. But when the grant ended, basically people folded up their tents and went their separate ways. Now there have been 2 follow-up grants to this original TCAP grant. The second one is just getting started. But they are completely focused really on breeding and genetics. And I've come to believe that we need a more collaborative effort. So this is just to give you a sense of what's going on in this arena these days. So here's 3 papers. This one from TAG. It's a good read, very well written. These folks tend to embrace the business as usual model more than I do. But it's a review article, basically. This other article, also in theoretical and applied genetics, certain to put a smile on the faces of Doctors McNear and McGrail, suggests that root systems are a logical breeding target. But again, it's a review paper. This is a paper where some actual work is being done in the field by a very innovative guy at Kansas State University, Krishna Jagadesh, who's developed these heat tents, and he's screened a lot of sorghum and wheat genotypes. And he's run the whole thing off of one of these tiny Raspberry Pi $100 computers. Very neat stuff. But the point is that a lot of people are writing about this and about the need for it, and here's what we should do with plant breeding and so on. Very few people are actually doing anything. And I think, it's difficult to do. It's very difficult to do on top of the breeding program and do it well. But as I said, I've come to the conclusion that it can't be divorced from other activities. Now, there are a number of available genetic and genomic tools that could be brought to bear on this topic. Genomic selection. You've heard Virginia and Jesse Carmack talk about genomic selection in their symposia presentations and their exit seminars. Basically, it allows one to screen tens of thousands of genotypes on the computer. I think that could be extremely useful in screening for resilience. We could employ reverse genetics. Plant breeders use forward genetics. We see an unusual phenotype, and then we do some experiments to try and understand the genetics. With reverse genetics, we postulate a gene that would be involved in resilience, we induce a mutation, and then we study the phenotype and see if it's one that might be favorable in a warming environment. And of course, genome editing is another tool. But again, I don't think this should be done in a vacuum of plant breeding and genetics. To me, the most exciting thing that's going on is this business of marrying crop growth models and genomic prediction models. And I don't think anybody's doing a better job of this than Charlie Messina and his group at Corteva. They're doing some very innovative work. And I think that there's huge potential here to harness these models and really identify what we need in the way of resilient genotypes. So it's in that spirit that I will mention this meeting that Yoko Kusanosi and Jairus Rossi, they're both in Ag Econ, and I are hosting in just about a week's time. We've been thwarted twice by COVID. The plan was to have an in-person meeting, and it's going to be virtual, for better or worse. We got a handful of breeders, a handful of ag economists, a smattering of physiologists, crop modelers, geneticists, climatologists, and basically we're just going to see what bubbles up in the way of collaborative ideas. Because I think that the breeding genetics effort needs to go forward, but it should be part of something that's much, much larger. Okay, so I want to thank these people, but there's one, there we go. I need to say thanks to some folks. First, the wheat breeding team. Pictured here, this past and present, for their ferocious work ethic and their unrelenting curiosity. and to Jim and Rebecca for graciously welcoming Katie and Caitlin into their lab so they could do part of their work there. And to the recently retired James Dollarhide, whose craftsmanship in devising the infrastructure for the passive warming study was just nothing short of amazing. These funding agencies, NIFA, Kentucky Small Grain Growers Association, and the U.S. Wheat and Barley Scab Initiative. And most of all of you folks for your kind attention. And with that, I'll be happy to entertain questions. Thank you.
Any questions? And also you can put questions in a chat box.
Is related to if you think that when breeding, if a breeder has a very active breeding for resilience, it can be like a slowdown in breeding for yield performance, or for yield, grain yield, and for characteristics of a variety in a very high performing environment.
Yeah, it's a great question, Vivi. And I think that if you carried out selection for resilience as part of your normal breeding program, there might be a negative impact on yield. I can't say for sure, I don't know. But I think that basically breeders have a day job, and that is breeding varieties for the here and now, right? And so the selection for resilience needs to occur separate from that. But it's a good question. I've wondered about phenotypic plasticity, something we tend to associate with genotypic or genotype by environment interaction, and we avoid like the plague. But maybe that's what we want, something that has that plasticity. Would it have a lower yield? I don't know.
Any more?
What are the implications that the SNPs for disease severity were found in your control environment, but you want to breed in a warming environment?
Well, they're not what we were hoping to see, Rebecca. So, you know, that would suggest that they're not going to be terribly effective in the warm environment. And, you know, this is not, I mean, we can do, and one should do, other searches for lines and germplasm that would be a source of disease-reducing genes in a warm environment. This was one panel, admittedly a diverse one, but I think that's what it means.
Since there's the lag between making the initial crosses and doing the selection and the variety becoming utilized by farmers, have you noticed, or is there any trend in varieties from far developed by breeders to the south of Kentucky performing better in Kentucky than varieties bred to the north of Kentucky? I don't know if you've thought about that or seen that.
Now that's a good question. And in fact, you know, back several years ago when commodity prices were really high and so on and so forth, people were entering varieties in Bruning's variety trial, you know, that came out of Louisiana and so on. And a lot of them did pretty darn well. Now, you know, that's not any kind of a scientific study, but just an anecdotal observation. Yeah, there have been a number of genotypes that have done quite well that were developed south of here.
Sure. So Dave, it seems like the photosensitive might be interesting to return to. So you talked about the polar vortex, and another aspect of climate change is probably that we're going to see these later freezes and that they may come even after unusually warm winters. So
exactly.
In terms of thinking about just all of it taken together, do you think moving towards the photoperiod sensitive I would assume they would fare better in a polar vortex late spring freeze too, is that correct?
Absolutely, yeah. No, it's a great question, and definitely, so we've got good markers for that, and when we can, when we're selecting progeny that are photoperiod sensitive, I think it's the way to go. I don't see a real downside to it. Breeders selected, in selecting for early maturity, one of the byproducts of that was they ended up with these insensitive types. But we can find plenty of photoperiod sensitive types that still head early enough to just do fine with double cropping and so on in our environment. So yeah, I think that would be a very sensible move.
And did you look in Lisa's study for the Dawn and the kernel? Was there any difference in sensitive and insensitive there?
You know, we didn't have as much marker data on the photoperiod loci, and that, I mean, they had a lot of genome-wide markers, but not necessarily QTL markers. So we talked about it, but we didn't have a good enough array of lines to really compare.
The presentation. I have a question regarding the performance of the photoperiod sensitive lines. So I was wondering if you were to use those on relatively cool years, what would be if you expect a negative trade-off? I was wondering if maybe those years that are relatively cool, they will induce to reproductive stages at the time that they have not produced enough biomass, and that will be negative. And another question related to that, I was wondering then if insensitive lines that have a higher thermal time will adapt maybe also well to high temperature without that possible negative effect under relatively cool years.
Yeah, interesting, very interesting question, Monty. So I think even during cold years and cool springs and so on, the photoperiod sensitive types are still producing biomass, okay? It's just that they have not elongated. So they're still tillering like crazy. There's still a lot of potential biomass. Now, I suppose if it warmed up really rapidly, then yes, there would be a yield penalty for the fact that they had not headed earlier. But the thing is, it's a huge safety, it's a built-in safety measure for these spring freezes that in 2020, they killed us. We had 2 killing freezes, and one in April, one in May. And so that alone is one strong point about that. Now, as to the other question, whether the insensitive genotypes might really flourish in warming conditions. Yeah, I think they might. I think maybe that is the way to go south of here. I think any place, though, that's got this prospect of spring freeze, I'm ready to go completely photoperiod sensitive, you know, because they can be devastating.
I was surprised about some results you show where the underwarming, you see earlier time of developmental stages, both anthesis and physiological maturity, but you have higher biomass, total biomass, but lower similar yield, I believe. So I'm wondering where is that extra biomass accumulating? It must be during seed field, because reproductive stages are occurring earlier. So are you seeing under that warming that there's more allocation to vegetative during seed field, since wheat is what we know is zinc limited?
I mean, we haven't done any partitioning work to validate that idea or to explore it, Monty. I know what you're saying. I think that we do in Kentucky have environments where we'll hit a period where the temperature is moderate enough that there will still be tillering at the same time that other later tillers are starting to fill grain. And if the season continues to be mild, then those later tillers will go ahead and make grain as well. So, yeah.
Okay, so I have a couple chat box questions, so maybe I should go couple them.
Okay.
So Wayland asked, what are your perspectives about the combination effects of new varieties and sustainable practices, e.g. cover crops, to cope with warming, drought, and pests? Any plan for such field experiments?
That is a good question, and I think speaks to this issue of collaboration and, not just doing breeding and genetics in a silo or whatever. And so working with folks on management. Yeah, I think it's a great question. I don't have any data to this at this point, but it's something that we should look at.
Thank you. And Yoko Kusunose, you have a list already of the types of research that must take place, but should this research be undertaken by crop breeders such as yourself in LGUs or by others like in ARS?
Right. So I think everybody should join the party, Yeah, there's a certainly in a perfect world, the ARS geneticists develop germplasm. So they would do the screening. They would develop the resilient germplasm, and then breeders would take it and develop varieties from it.
And Bob Pierce, how long do you think the time frame would need to be for selection for resilience traits? Seems that the typical time frame for selection might be too short.
Yeah, I guess it would depend on the trait and the protocol that we were able to develop screening for it. That's a good question. We're always looking for a trait or a proxy that is efficient and that we can run big numbers with. But it may be too short. I just don't know at this point.
I think we'll take the last question.
Thank you. Congratulations, Dave, on this very successful work. And on your ongoing efforts now, these multidisciplinary concerted efforts. I'm just curious, in your opinion, what has soil science not delivered yet, and what could soil science deliver to make this concerted effort even a little more successful?
Right. So, when you mention soil science, when I've thought about this, I have thought about these folks that hang out in the rhizosphere and look at roots. So I think that would be a good place to start. It makes sense. Terribly difficult research to do, but it makes a heck of a lot of sense to look there. And then, I think I mentioned that one fairly recent modeling paper, they cited among their traits that could be combined into a resilience package, early water use efficiency. And so I think that would be another area, Ola, soil water. Yeah.
All right, thank you very much. I'd like to thank again Dr. Dave Van Sanford for giving us your great project talk. Thank you.